In this video, I will be discussing the process of a developmental network that leads to the formation of spikes in a dragon. These are genes that lead to the transcription of spikes in the dragon's skin. In this case, gene S activates gene P, P represses I represses K activates E, and E activates the transcription of spikes. To better understand how a developmental network affects gene expression, I'll be using an analogy. Let's say you're a farmer trying to get your sheep across a bridge to your farm safely, but a fox blocks your path. In this case, transcription of spikes is equivalent to our sheep being safe. So for this network, each of these equates to one of the genes in our dragon's network. Having a whistle calls your dog, the dog chases the fox away, the fox blocks your path through the bridge ensures a path to the farm, and the farm ensures the sheep's safety. Now let's look at different strains in our networks to see if they activate transcription. First one is wild type, which is the normal phenotype that occurs naturally. Wild type will activate spikes, or in our analogy, the sheep are safe because of the specific genes that are activated and repressed. It is also given that this network is meant to transcribe spikes. Next is gene P, which will also produce spikes. P is responsible for repressing gene I, which stops other activator genes and thus stops transcription. Gene P ensures that spikes form, equating to the dog chasing the fox away, providing a clear path for the sheep. Next up we have gene I. Gene I will not produce spikes because it represses gene K, and since gene K is responsible for activating gene E, which forms spikes, having gene I will cause spikes not to form. And since having our fox blocks our path through the bridge to the farm, our sheep aren't safe. For I and E, these will not produce spikes. Although gene E is what activates transcription, having gene I represses gene K, which is needed to activate gene E. So although the farm ensures our sheep are safe, with the fox in the way, we won't be able to get there anyway. Finally, we have S and K, which will produce spikes because they are both activators in transcription. This also means that having a whistle calls our dog to chase the fox away, and the farm ensures the sheep are safe. Now, even though this network transcribes for spikes, not all of our cells will have spikes. This is because of an epigenetic process called methylation. Methylation can decrease transcription by tightening the area between the DNA and histone. Tightening changes which parts of the DNA sequence are a part of the nucleosome or a part of the area in between the nucleosome. Doing this affects how exposed a part of a DNA sequence is. If a part of a sequence is less exposed, an activator or repressor protein is less likely to bind to that transcription site. Some cells may have tighter DNA than others, resulting in some cells forming spikes and others not.